Hello, my name is Dr. Roderick L. Roll, and today I will be talking about chromosomal abnormalities. Chromosomal abnormalities can be further divided into two categories: chromosome number alteration and chromosome structure alteration. Large-scale chromosomal alterations in humans and other mammals often lead to spontaneous abortions, which are also called miscarriages, or you can cause a variety of developmental disorders. The CDC suggests that if you lose the developing embryo by week 20, it is called a miscarriage. And after week 20 up until week 36, it's called a stillbirth. One percent of all pregnancies end in stillbirth. During nondisjunction, members of a chromosome pair fail to separate during either anaphase 1 or anaphase 2. If this occurs, a gamete will be created that has an incorrect number of chromosomes. In this slide, nondisjunction took place. Spindle fiber will either break or it will not form. So in this image, the tetrad is supposed to be pulled apart, but for some reason it did not. That reason is either the spindle fiber broke or it did not form. In this image, the chromosome did not separate. Either the spindle fiber did not form or the spindle fiber broke. In the first scenario, where anaphase 1 was the culprit, nondisjunction of homologous chromosomes will not sever. This error is created in meiosis 1. In that case, look at the end result. You'll have two gametes that will have an extra chromosome and two gametes that will be minus a chromosome, N minus 1. In the second scenario, nondisjunction of sister chromatids, that occurs in meiosis 2. If the sister chromatids are not pulled apart, that error will create 50% of the gametes will be damaged. So one gamete will have an extra chromosome while the other gamete will be minus a chromosome. But in this scenario where nondisjunction takes place in meiosis 2 instead of 1, you'll at least have 50% of the gametes will be normal. Aneuploidy results from the fertilization of gametes in which nondisjunction occurs. This is a condition of having an abnormal number of a particular chromosome. You will either have an extra chromosome or minus a chromosome. If you have minus a chromosome, as in the previous images, you will be monosomic. If you have an extra chromosome, you'll be trisomic. So monosomic zygotes will have one copy of a particular chromosome. An example is Turner syndrome. This female will have one X, and she is supposed to have two X's. A trisomic zygote has 3 copies of a particular chromosome. In the example, we're looking at Down syndrome. That individual will have a extra chromosome number 21. In the next couple of slides, I'm going to show you a karyotype, and I would like for you to tell me if this individual is a male or female, and if he or she is normal. So based on this karyotype, if you look at every chromosome pair, this individual is normal, and he is a male because he has a Y chromosome. If we look at this karyotype, all chromosome pairs are there, so this is a normal female. This individual here has extra chromosomes under the X category. So this person has 4 X chromosomes and they have a Y chromosome. So this is a male, but he is abnormal. He has what is called Klinefelter's syndrome. So Klinefelter syndrome has poor beard growth, narrow shoulders, wide hips, long legs, small testicles, female type pubic hair pattern, breast development, and so on. Now look at this individual and tell me, is this person normal or abnormal? This person is abnormal. How are they abnormal? Very good. She has an extra chromosome, chromosome number 21. She would have Down syndrome, a flattened facial features, small head, short neck, and so on. What is this? This individual is a female and she is abnormal. She has an extra chromosome number 13. That is Pate's syndrome. Small head, absent eyebrows, cleft lip or palate, extra fingers, polydactylity. What is this? Yes, this is a female, but she is abnormal. She has an extra chromosome number 18. That is Edwards syndrome. So this individual has heart defects, small abnormally shaped head, small jaw and mouth, and clenched fist with overlapping fingers. 5 to 10% of children with this condition live past their first year. What is this? Yes, this individual is a female, but she doesn't have 2 sex chromosomes. She only has 1. She has Turner's syndrome. Turner syndrome is a shortened height, failure to start puberty, so she's infertile, and she has heart defects. What is this? This individual is a male and he is not normal because he has an extra Y chromosome. He has Jacob's syndrome. The affected individuals are usually very tall. This is the characteristics of Jacob's syndrome. Examples of syndromes that survive to birth are as listed. Klinefelter's syndrome has XXY. Metafemale has extra X's. Turner syndrome has XO. And Jacob's syndrome has an extra Y, XYY. So all of those are defects in the chromosomal number, and all of those are in reference to the sex chromosomes. To the far right, these are autosomal abnormalities. They have extra chromosomes that are in the category of autosome. So Down syndrome has an extra 21. Pate syndrome has an extra 13. And Edwards syndrome has an extra 18. These are examples of syndromes that survive to birth. Most of the other abnormalities with chromosome numbers do not survive. You're going to be either miscarried or you're going to have a stillbirth. So we want to look to see who is at fault. Both parents are at fault. In this slide, we're showing you that Klinefelter syndrome is going to be seen 1 out of every 2,000 individuals. And what happens is either the egg has the abnormal chromosomal count or the sperm has the abnormal count. To create a Jacob's individual, the male is at fault. Superfemale, either the egg malfunctioned or the sperm malfunctioned. And then with Turner, it's either the egg or the sperm. This graph here shows the importance of not waiting. So they did a study and looked at the average age of a mother that has a child with Down syndrome. As you can see, in the early 20s, the females that create babies have a 1 out of 2,200 chance of creating a baby with Down syndrome. Let's look at age 25. Age 25, it increases to about 1 out of 1,600. Age 35, about 1 out of 290. And by the ripe age of 40, 1 out of 100 babies will have Down syndrome that were produced from women that were 40 years old. So as a woman ages, her chances of having a Down syndrome individual increases. This means that nondisjunction abnormalities increase as you age. This information was taken from a very reputable website, which is sponsored by the National Institutes of Health. And it says it is possible that one aspect of the strong maternal age effect on nondisjunction is an age-dependent decrease in the probability of keeping the chromosome tetrad together. So, especially during a female's life cycle, her tetrads are held in place until menstruation. Once the menstrual cycle occurs, one developing egg is going to be activated to continue the meiosis process. So what happens is this egg, developing egg, was suspended and frozen in meiosis. So this is why we have so many defects in reference to women. The tetrad is being held by the centromeres, by the spindle fibers, and it is held there from when she's born until the day that she goes through menstrual cycle. Then one of those developing eggs will be jump started, and you will then separate the tetrad. So let's look at a picture to better explain this. In this slide, you see that in the red highlighted box, I have the primary oocyte, which arrests in prophase 1. This occurs at birth for every female. So at birth, all of her potential eggs are frozen in time. Then go to the next box. It says secondary oocyte, arrest in metaphase 2. So once a month, once she reaches a certain age, a woman is going to jumpstart that forming egg that was suspended in prophase 1, and she will go through metaphase 1, anaphase 1, telophase 1. Then she will go into prophase 2 and then metaphase 2. Once it reaches metaphase 2, she releases the developing mass of cells. When she releases that mass of cells, that is referred to as ovulation. And that occurs once a month. So only one of those growing cells is going to be activated or jump started. Then, luckily, if sperm is present, then meiosis will continue and go from metaphase 2 to anaphase 2, then to telophase 2. And then once it reaches that, then the sperm is able to penetrate the growing egg, break down the zona pellucida, which is a outer shell, and then be fertilized. Let's look at alterations in the chromosome structure. So we just completed looking at alterations in numbers and what that will cause in the human population. Now let's look at what happens if pieces of the chromosome break off. Breakage of a chromosome can lead to 4 types of changes in the chromosome structure. In this image, we're looking at some breakage that occurred during anaphase. The first abnormality that we'll look at is a deletion. So deletion removes a segment of the chromosome. In this image, we're going to take gene D and we're going to delete it from that chromosome. So whatever gene that was, we're no longer going to produce the proteins that were from it. This could actually lead to death in the developing embryo. One example is Cry-Du-Chat syndrome. This syndrome results from a specific deletion in chromosome number 5. If you look at the image below, the piece of chromosome 5 has been deleted. A child born with this syndrome is severely intellectually disabled and has a cat-like cry. Individuals usually die in infancy or early childhood. Duplication is another example. This occurs when you have a repeat of different genes within a chromosome. In the image below, we're looking at gene B and gene C. Notice that they have been duplicated. So when you duplicate a small piece, such as on chromosome 1, this can lead to macrocephaly. It can also lead to developmental delays, learning difficulties, autism, heart problems, seizures, and distinctive facial features. So deletion and duplications are typically tied in together. This occurs during meiosis. Let's look at the image to better explain this. In prophase 1, when a tetrad forms, notice that the two non-sister chromatids are very close together. If we follow number 2, we have gene A, gene B, then gene D, E, and F. Notice that E is not in that number 2 chromosome. Then number 3 chromosome is A, B, C, C, D, E, F. So when that crossing over event occurred, some error occurred. And one of the genes were deleted off of chromosome 2, but it was added to chromosome 3. So whenever you whenever you have a deletion, you will also have a duplication. In that case, if the gamete that was formed had chromosome 1 in it, you would be normal. If it had chromosome 4, you would be normal. But if it has either 2 or 3, something is wrong. The next one we'll look at is inversion. This is when the arrangement of genes will be reversed. So instead of having gene B, C, D, you're going to have D, C, B. The fourth one is translocation. Translocation moves a segment from one chromosome to a non-homologous chromosome. Notice in this image, the genes A and B are going to be taken off of chromosome 1 and put onto chromosome 15. And chromosome 15 has gene M and O. Those genes are going to be translocated to chromosome 1. Certain cancers, including chronic myelogenous leukemia, are caused by translocation events. If you switch a section of chromosome 9 with a section of chromosome 22, you create this type of cancer. This is just a summary of all of the chromosomal alterations.